Greetings, everyone. I would like to start by once again welcoming you all for this webinar. I'm very excited and I thank the organizers for giving us the opportunity to discuss regulatory strategies in the field of cell and gene therapy. Karen and I both work for Lonza, and this is our disclosure slide. This slide has a brief overview of Lonza. Lonza is a global leader and well-known CDMO in the field of life sciences. Lonza was founded in 1897, and we have more than 50 manufacturing sites worldwide in more than 30 countries with close to 15,000 employees. At Lonza, our mission is to industrialize manufacturing from concept to patient, and within that scope, we focus on drug development, manufacturing drugs and biologicals for both clinical trials and mature market supply. We have a strong presence not only in chemical technologies and biologics in general, but also in emerging technologies such as cell and gene therapies. Lonza's vision for our two key therapy areas of cell and gene therapy is to deliver the medicines of tomorrow today. How we do it is by having a global footprint and flexible capacity, and through innovating in key areas such as development and manufacturing. In the US, we have manufacturing sites for cell and gene therapy at Portsmouth and Houston. We also have sites in Netherlands and Singapore, and also a partner site in Japan, which is owned and operated by Nikon. For today's webinar, we would like to discuss the existing regulatory landscape, guidance, and regulations applicable to cell and gene therapy. We will touch upon a few hot topics related to manufacturing facilities to be considered during filings, and also Lonza's regulatory capabilities and offerings such as master files that sponsors can utilize to accelerate their regulatory timeline. Now I will pass it on to Karen to continue.
Thank you, Raj. We're now going to transition into a discussion of some of the regulatory hot topics that are relevant for the manufacturing of cell and gene therapy products. This next slide contains a great deal of information and is not intended to overwhelm you. It is a visual representation of the timeline of the emergence of cell and gene therapy relevant regulations and associated guidelines and guidance documents that interpret the regulations. This is a complex regulatory framework for these types of products, and this is only a handful of the relevant documents. Back in 1984, the FDA began to regulate gene therapy products, and since then, Japan and Europe and other regions have also laid down their regulatory framework. This framework continues to evolve as guidance documents and guidelines that were issued early on have now been up for revision, many of them, and continue to be revised on a rolling basis as more information is gained in terms of the technologies involved for these products and also more experience in the development pathways for these products. There is one guidance document that I'd like to highlight in the center of the slide. It may seem to be an outlier when grouped in with other cell and gene therapy regulations and guidance documents. It is the content and format of chemistry, manufacturing controls information, and establishment description and information for a vaccine or related product. I'll be circling back to that particular guidance document later in this presentation. There are two documents that have been fairly recently issued that are very important documents when beginning to understand the regulatory framework for these products. And I am pulling out and highlighting these two because if you are new to this or wanting to take a deeper dive into the regulations and interpretations of the regulations for these products, these are two must-read documents. The first was adopted by the European Commission in November of 2017 and went into effect in May of 2018. It is the Guidelines on Good Manufacturing Practice specific to Advanced Therapy Medicinal Products, or ATMPs. This document is highly prescriptive in the practices that should be followed when manufacturing these products. It lays out potential processing scenarios that are based on the tenets of segregation in time or segregation in place for prevention of contamination, cross-contamination, or mix-ups. Also has specific information about multi-product manufacturing, and again goes into many details of aspects of the GMPs. Likewise, in terms of level of detail, there was an excellent guidance document issued by the US FDA after an extensive public comment period. This was issued in January of this year. This is specific to chemistry manufacturing control, or CMC, information for human gene therapy investigational new drug application. So this lays out recommendations for the type of information to be submitted along the clinical development pathway and the format for how to present that information in an IND. This is specific to gene therapy products, but one can assume that many of the recommendations in this document would apply for applications for cell therapy products also. This slide contains quite a bit of regulatory jargon. It is because it is describing some of the regulatory pathways or designations or programs that are available to sponsors developing products, including cell and gene therapy products that are seeking an accelerated development pathway and hopefully an accelerated approval pathway. And I've just highlighted some from the US FDA and from Europe. The FDA, we've got breakthrough, fast track, accelerated approval, and the latest program to be issued, which is the Regenerative Medicine Advanced Therapy, or RMAT. These all have criteria that need to be met in order to be granted approval for that program. They all are based on serious conditions, but the criteria for demonstrating potential effectiveness of the product differs. These programs, many of them enable potential priority review of the commercial filing. Similarly, in Europe, there is a PRIME designation, again for serious condition or unmet medical need, a conditional marketing authorization, which is not a program available in the US, and again, similar to priority review, an accelerated assessment. One program that is not described on this slide, but many cell and gene therapy products have a designation, is orphan drug status. Orphan drug designation is based on the number of individuals treated with a product in any given year. And because many cell and gene therapy products do have orphan or ultra-orphan designations in terms of the numbers of individuals to be treated, that is a program that is being followed for many of these products. Because many of the cell and gene therapy products have one or several of the designations on the preceding slide, it does mean that one of the biggest challenges, overall challenges in the CMC space for these products is the timing of executing the CMC portion of a development program for an expedited program. So the overall intent is to ensure that therapies for serious conditions are available to patients as soon as possible. However, for many of these products, it means that clinical development timelines are accelerated in such a way that CMC and GMP issues are on the critical path. Regulators are out there publicly talking about the fact that there will be no quality compromises. This is evidenced, again, in the guidance documents and guidelines that have been issued, and even review memos from approved products illustrate the same concept. So with no quality compromises along the way, this means that sponsors need to work closely with health authorities so that all can understand and avail themselves of opportunities for flexibility, and there may be post-approval CMC efforts to be expected. So let's explore some of the regulatory hot topics, and I selected these based on things that regulators are out there speaking about in the public domain. Also, again, things that are highlighted in the guidance documents that have been issued, and also some specific advice that Lonza has obtained from regulators, either directly or as we support our customers in their journey towards commercialization. And as this audience certainly knows, the manufacturing of cell and gene therapy products is highly reliant on aseptic processing for the most part. As a result, many of these hot topics are aligned to some of the challenges around aseptic processing, and the first of which is cleaning. As many of you know, although much of the aseptic processing steps occur in closed systems, open steps do need to take place in biological safety cabinets. And so there should be no surprise that the cleaning of those BSCs is of interest to regulators. So proper cleaning and line clearance and how you do that between products and batches is of interest. Also, the demonstration of the effectiveness of cleaning programs. So things like disinfectant effectiveness studies, how they're designed, how they're executed, and the data is interpreted is of interest to health authorities. HVAC system and other support systems and environmental monitoring we'll be covering in more detail in subsequent slides. But before we leave this slide, new product introduction is something that, again, is of interest, especially as we all know that are on this call, there are many technologies that fall under the umbrella of cell and gene therapy. And one facility, one manufacturer may be handling products across different technological modalities. And even, for instance, if we just look at vectors and just even down to viral vectors, there may be different types of viral vectors for different types of programs that are being handled in a facility. And so a risk assessment needs to be performed every time a new product may be brought into that facility to assess whether or not the control strategy that is in place for the prevention of contamination, cross-contamination, and mix-ups is adequate for the new product to be introduced. And again, going back to the closed systems, this is a phrase that's often used in aseptic processing. The way that the closed system integrity is verified is through media simulation studies, and regulators are very interested in how these studies have been designed, how the aseptic process simulation maps to the actual manufacturing, and how the data is interpreted. Validation summary information is something that particularly the US FDA is showing a keen interest in information around equipment and utilities and computer systems for these types of processes, and requesting information to be submitted in commercial filings regarding the validation of, again, especially critical equipment, utilities, and computer systems. Personnel, material, and process flows I'm going to talk about a little bit later, but an emerging hot topic is manufacturing capacity. And this seems to be emerging in the context of autologous processes for which scale out becomes relevant for commercial product, wherein in the clinical phase, there may have been a limited number of batches produced, and maybe one on a day in a particular suite. As the ramp up to commercial occurs, scale out means that there may be parallel processing of multiple batches in a single suite. It may mean that there are multiple production suites operating within a facility. And so particularly the US FDA has been making some recommendations in terms of considering not only the capacity in a production suite or set of suites, but also considering holistically bottlenecks in QC, logistics, facility equipment, and operators in the facility as a whole. And now to go back to that guidance document, the 1999 guidance document that was issued for vaccine or related product. And I entitled this slide, What is Old is New, because many of the tenets in this guidance document have followed through or are carried through in the way that cell and gene therapy manufacturing establishments are evaluated. And the FDA is still citing this guidance. And we see questions and comments and recommendations come from the FDA that are perfectly aligned to this guidance document. So some of the concepts for manufacturing these products, even though there's novel technologies, the underlying principles have not changed for how a manufacturing facility should be operating. So some of the highlights from this guidance document, again, are aligned to some of the hot topics. New products in the facility, a list of other products manufactured in the facility is something that health authorities are very interested in. The building of floor diagrams indicating the facility layout. This is something that needs to be easily readable, potentially manipulatable, because authorities are very interested in the product, personnel, equipment, waste, and airflows, and any intersections or risks, especially in a multi-product facility. The HVAC system, again, this is something of keen interest in maintaining that aseptic processing manufacturing environment. This guidance document gives some specific details in terms of the information to be provided for the HVAC system, the number and segregation of the air handling units, whether the air is once through or recirculated, containment features, air changes per hour, air pressure differentials between adjacent areas are all things that are requested to be in the HVAC description. The environmental monitoring program is something, again, of keen interest in an aseptic manufacturing environment. It is of keen interest to health authorities, I should say. So things like the tests performed, the viable and non-viable particulate action and alert limits, and even the justification for the acceptance criteria should be described within a filing. And as I mentioned earlier, the water systems, computer systems, are of interest to regulators, and providing details around the validation for those systems is also important. Contamination and cross-contamination issues. This is something that in that CMC guidance document for gene therapy, the type of information that should be submitted, this is mentioned in that guidance document. It's certainly mentioned quite a bit in the guideline for ATMPs, for GMPs. So not only considering contamination, cross-contamination prevention and control strategies from very beginning of manufacturing, there also needs to be a consideration for how this is described in relevant filings. And now back to Raj.
Thank you, Karen, for setting the stage well for me to explain our offerings and our regulatory support mechanism for sponsors. Just to give an overview, in the last 6 years, we have supported more than 60 clinical trial applications and more than 12 marketing applications in the biologics field in particular. We have the capability to provide regulatory support tailored to any of the stages involved in the drug development. As we are a CDMO, our clients span across the spectrum between preclinical early stage clients to sponsors gearing towards phase 2 or 3 and to license application and to even commercial clients. Therefore, we have the resources and required team to support our clients during all their phases. We provide strategy support in terms of planning and supporting regulatory aspects during the early product development. We also support sponsors during the clinical trial application phase and also provide a comprehensive support during the license application and during the post-marketing authorization phase. I will discuss this in detail in the following slides. Our regulatory support include regulatory recommendation and strategy development during process development and technology transfer for CGMP manufacturing. This is especially relevant and very well appreciated by our early stage clients. We have experience to navigate the regulatory landscape around import-export of critical raw materials, animal origin materials, and drug substance and drug products. We work closely with our clients and local and federal regulatory agencies to procure the essential permits and licenses as needed to ensure compliance. Each of our sponsors receive dedicated regulatory representative to support their project. The regulatory affairs personnel will work closely both with the internal Lonza team and the client's team to provide either strategic or technical support. Few examples include providing support to write briefing documents, participating and contributing during regulatory authority meetings, and responding to information requests from the regulatory agency during their application review process. Post-approval support include providing annual product review, master file updates, and also to help with the post-approval changes. We provide all our regulatory documentation in common technical document format, and we have tools and systems in place to perform electronic submissions to relevant regulatory agencies. For our clinical and commercial clients, we support either drafting or review of the quality section or module 3 of the filing, including the advantageous agent safety evaluation section. We typically draft the facility and equipment section for our clients' marketing application process. We also have a lot of experience in writing and maintaining master files, which I will explain in more detail in the next few slides. Drug master file is a regulatory mechanism to provide companies confidential and proprietary information directly to the FDA. These are some applicable regulations which states that master files can be used to support clinical and commercial applications. Some examples of confidential information include facility details, process details or articles used in the manufacturing, processing, packaging, and storage of drug substance or products. Risk evaluation and mitigation strategy and also toxicology information can also be provided in a drug master file. Master file can only be used to support sponsor's application and does not replace or substitute a sponsor's application. Master file holder authorizes one or more applicants or sponsors to cross-reference the information available in the master file. FDA neither reviews nor approves a master file submission independently, and it is always reviewed in the context of a sponsor's application. Needless to say, the contents in the master file are not disclosed to the sponsors. These are the different types of master files, and they are listed here. Type 2 master file is for drug substance and intermediates. For BLA applicant, the FDA expects the information in the Type 2 master file to be submitted directly to the BLA though. Type 3 master file is for packaging material, and Type 4 master file is for excipient. Type 5 master file is for any other FDA accepted information that does not fit into the other types. For example, it could be REMS or information around a sterile processing facility or toxicology studies. To submit a Type 5 master file, the DMF holder has to file a letter of intent with the FDA. This slide shows the count of DMF by the type that different business unit of Lonza holds. Our cell and gene therapy business unit has both Type 2 and Type 5 master file, but majority of them are Type 5. In our master files, we currently support and contain manufacturing site information, cell banks, and its manufacturing processes. We are planning to have master files that would include information on single-use systems, manufacturing platforms, and processes for viral vector production and for producer cell lines. Our master file typically will not cover any product-specific details such as potency or stability assays. This is how a typical process flow looks for a master file cross-reference procedure. The master file holder submits the master file to the FDA. The sponsor requests the master file holder for letter of authorization. The master file holder in turn submits a letter of authorization to the FDA and also provides a right to refer letter to the sponsor. Finally, the sponsor files the right to refer letter in the corresponding filing to complete the process. Let's now see how the master file submission process with the FDA works. Before the submission, we need to procure a pre-assigned DMF number from the appropriate center. For a CDER master file, the request can be submitted via online portal, whereas for a CBER master file, the request can be emailed to the address provided here. DMF has to be submitted electronically, and files 10 GB or smaller must be submitted through the electronic secure gateway. Submission over 10 GB can be either submitted through ESG or on a physical media like a CD. I don't want to go into the details, but depending on the type of the master file, the CTD section requirement may vary, and relevant content should be provided in appropriate modules. Subsequent submission for a DMF could be anywhere from filing a change in contact or changes to DMF title or type, or just issuing an LOA or providing quality amendments. Annual reports are very important, and they help assure FDA that the information in the master file is current. Annual report typically contains a cover letter, a statement signed by the master file holder stating that the master file is current. The annual report should also list the dates of any amendments, status of the administrative information and changes, if any, and an updated list of authorized parties. Failure to submit this report annually may result in termination of the master file. This is a snapshot of a template for the annual report. This is our final slide, and it shows how we keep abreast with the evolving regulatory guidelines and shape a regulatory framework for cell and gene therapy products. We review and provide comments on draft guidelines that are being published by regulatory authorities. We actively participate in industry groups such as Alliance for Regenerative Medicines, BPOG, CAS, and Standard Coordinating Body, to name a few. We also do present our innovations to the CBER Cell Therapy Working Group and also support and contribute to the EDQM Gene Therapy Expert Working Group. Thank you for your attention, and we once again thank the organizer for this opportunity.